Hi everyone, I'm Maiko and I'm a grad student at UC Berkeley, and I'm here to tell you about my project looking at chromosome segregation defects in Xenopus frogs. At some point, every single cell in your body has gone through a division process, where one single cell has to become two absolutely identical daughter cells. This means that all of your genomic information has to be replicated and then segregated faithfully into these two cells. Through evolution, people have noticed that many hybrids have trouble with this process and lose some amount of genomic material. Maybe it's a single chromosome, maybe it's the whole genome from a particular parent. Now, I'm studying this with various frog species because if I fertilize them in a particular way, I can get hybrids that actually die during development. They don't make it beyond the first early embryonic divisions. When we looked subcellularly, we found a lot of chromosome missegregation defects and micronuclei formation. And when we use sequencing technology, we can see specifically which chromosomes are affected. What we found was very striking. First, only the paternal chromosomes are affected and lost during each of these embryonic divisions. And secondly, the missegregation is actually really specific to only a subset of chromosomes. However, we have no idea what about these chromosomes predisposes them to be missegregated. What makes them different from all of the other chromosomes in the paternal genome? And so for my project, I'm looking to see what particular incompatibilities may exist between the two species that make up our hybrid. Now, during cell division, when our chromosomes are condensing into these rod-like structures, they have a very specific region near the center called the centromere. Now, the centromere is essential for division because this is the part that attaches the chromosome to the division machinery, namely the microtubules of the mitotic spindle, which will then pull them apart to the relevant daughter cells. However, it's been known that through evolution, the centromere is actually a much more highly divergent region of DNA compared to the rest of the genome. And as a result, the proteins that are necessary in specifying and assembling the centromere are also much more divergent, even across very closely related species. And so for my work, I'm looking to see if I can supply the maternal egg with paternal-specific centromere factors. Will that save these paternal chromosomes from getting missegregated or not? And so not only does my project look at the mechanisms of how the centromere helps maintain faithful segregation, but I'm also adding an evolutionary aspect to it by providing different species of proteins in order to either help or hinder this process. What's actually very interesting in using the Xenopus species is that we can also use a third species to create a different hybrid. This hybrid actually does not die in the same way, but has the same timing of death as our first original hybrid. Now, this hybrid actually also undergoes chromosome missegregation, but actually the chromosome loss is a little bit different. Again, only the paternal chromosomes are lost, but the extent to which these chromosomes are affected is actually much less, and only bits and pieces of specific chromosomes are lost, as opposed to whole chromosomes in our original hybrid. And so this begs sort of a different question. What is different about these other chromosomes that leads to their fragmentation as opposed to their entire loss? And so when we tried addressing potential centromere incompatibilities with this second hybrid, we actually found that maybe these mechanisms are, the mechanisms of loss are actually different, simply because the centromeric factors did not seem to have an effect on the second hybrid. And so this also begs the question, what other mechanisms might be involved in mediating chromosome breakage and segregation defects during the early embryonic divisions? Now, I don't have an answer yet since I'm still working on the project, but this provides a great context to look at both an evolutionary standpoint to see how these mechanisms might be conserved across species and how they might change depending on the situation, i.e. depending on which species is fertilizing which other species. And also this provides an interesting window into how speciation occurs. Speciation is more or less defined as reproductive isolation, where two species after mating together cannot produce progeny that can then propagate and create more organisms. And so what we see is a early embryonic death, which is actually a very interesting way of preventing these two species from mating. And so in conclusion, I hope that my project will shed some evolutionary light on a very key and very important process, which is cell division, which occurs in all of our cells. And I hope to find something interesting about the specificity of chromosome missegregation and why specific chromosomes might be more or less inclined to become missegregated. Thank you.